Okay, this is part 11 and the last material for this week. Moving on to page 356, section 13.10, other infectious agents, viroids and prions. So the first one we're going to mention are viroids. Just to say what they are, and really just to say that we really don't know. There's more questions than answers about viroids. Viroids are small, single-stranded RNA molecules, and that's all there is to them. There are no proteins involved, there's no DNA involved, there's small, single-stranded RNA. About 200 to 300 and a bit nucleotides big. So a nucleotide is one of the letters. So in terms of RNA, it's A's, U's, G's and T's. That size of RNA molecule is about 1/10 the size of the smallest known RNA viruses themselves. Viroids form these closed ring structure that can often give the virus a double-stranded look. It looks double-stranded, but it's not. Currently, we only know of viroids as being single-stranded. And so far, we only are aware of viroids causing, or indeed being found, in plants. So viroids do cause disease in plants. I was just looking to see if there was any more current information about viroids and disease. There is looks like a beginning debate or beginning questions into the difference between the hepatitis D virus and a viroid. Some claiming that hepatitis D is indeed a viroid, some just saying that hepatitis D is a RNA virus, but it is very similar. So there's not, as it seems, as with most things, there's not one kind of thing that is a virus and one kind of thing that is a viroid. Likely, as we investigate these kinds of things, we're going to find instances of these acellular infectious agents that are kind of partway between. But we are going to leave it at the idea that we just don't know a lot about these things. How do they replicate? How do they cause disease? RNA molecules in these viroids are not known to be able to encode for proteins, for example. Where do they come from? Do indeed viroid counterparts that cause viruses in plants have counterparts in animals? That's where we'll leave it. Things that we do know cause disease in animals are prions. And prions stands for proteinaceous infectious agents. So they are only proteins, no nucleic acids. They are linked to slow, currently fatal human diseases and also animal diseases. Currently fatal in that once you are infected with a prion, we have no known cure. Prions are notoriously difficult to treat. They can't be killed by intense heat. They are resistant to most chemicals. So just one problem in combating prions is just our inability to cause to harm them. And clearly, if we are going to attack a virus, a prion, with an extremely harsh chemical or extremely harsh conditions, and that prion is inside a human brain, then very unlikely we're going to be able to harm the prion without harming the human at the same time. All current known prion diseases are linked to neural tissue, and that's brain and nervous tissue. And there are some examples in the table on page 357. The one that you may have heard of affecting humans, Creutzfeldt-Jakob disease, can be passed between animals. There was a disease called mad cow disease. And there was a transmission of the prion in infected cow meat. So it's an unfortunate fact that it's very difficult to slaughter and prepare cow meat without also contaminating that meat with neural tissue. And there's a certain amount of neural tissue that's allowed in beef and in, say, in hamburger. And if you were to then slaughter and prepare the cow, and it was infected with mad cow disease, a prion disease, and that prion got into the meat, and that meat gets into the person, it doesn't matter about cooking your hamburger, that burger is going to be infectious. There's also, if you know hunters who kill and prepare, say, deer for consumption, there's a lot of may be aware of prions in deer that could potentially be transmitted to humans who eat that prepared meat. When a prion gets into neural tissue, say the brain, it causes this kind of characteristic appearance, this spongy appearance with holes in the brain, and that is why these diseases are generally termed transmissible spongiform encephalopathies. Let's take a look at how these infections occur.
Prions are hypothesized as the causative agents of several neurodegenerative diseases. Prion diseases are unusual because they appear to be caused by an infectious protein. The proposed mechanism of the disease is that the infectious protein catalyzes a conformational change in a normal protein so that it becomes the same as the infectious protein. According to the proposed mechanism, the normal protein is found in neurons in the PRPC conformation. In this conformation, the protein is harmless. The infectious protein conformation, PRPSC, is introduced to the neuron after the individual comes into contact with it in their food, or in rare cases from contact with bodily fluid of an infected individual. There are also some inherited cases where it appears that the PRPC can convert spontaneously to the PRPSC form at a low rate. However, the PRPSC form gets into the neuron. Once it is there, it contacts a PRPC protein and catalyzes its conversion to the PRPSC form. Once it is converted, it too can act to convert other protein molecules from the PRPC form to the PRPSC form. Over time, all of the PRPC proteins will be converted to the PRPSC form. Some PRPSC proteins are excreted from the cell and thus are able to spread to and infect other cells. PRPSC proteins accumulate to high levels over time. They are deposited as dense aggregates that are thought to be responsible for disease symptoms affecting the nervous system.
Okay, so we have, as a regular part of our proteins in our neurons, we have proteins who have a regular job to do. And this is another example of naming that isn't very helpful, because this prion protein sounds like it's a protein that's meant to become a prion, but clearly it's not. Once again, no cell wants to produce a dangerous protein. This protein has some other function, some regular function that is important to the function of the neuron. It just so happens that if the conformation, if the shape of the protein changes, then it becomes dangerous. It becomes changes from the normal PRPC to the abnormal PRPSC. The SC stands for scrapie. So PRPC stands for prion protein cellular, i.e. the cellular configuration. PRPSC stands for prion protein scrapie, the infectious form of the protein. And as you saw there, the regular protein that has an important function in the cell, it can itself be changed into the infectious agent. The infectious agent, the PRPSC, has a particular shape, and that shape can change the shape of the normal protein. And that is how it is hypothesized that the infection progresses from protein to protein and then from cell to cell. So we get the entry of the bad shape. The bad shape interacts with the regular proteins, changing their shape. And now we have more bad shapes, and they can interact with more proteins, and they can change their shapes until the majority, or indeed all, of the proteins are in this let's call it a disease configuration. And then it's thought that these disease configuration proteins form these fibers in the neural cell, and it's this fiber configuration that then disrupts the function of the neuron, killing the neuron and causing degradation of the neural tissue until you see these holes that are simply the result of this patch of neural tissue being killed by the accumulation of these prion proteins. So, what are viroids? Short pieces of single-stranded RNA, and that's it. We know that they cause diseases in plants. We're not quite sure whether they cause diseases in humans, and we're not quite sure about many of the aspects of viroids. There are also many aspects of prions we don't understand. Okay, so what are prions? What are the characteristics of human disease? Prions and disease always affect neurons. In humans, typically it's a slow disease, and in humans there is typically no known cure. And have an idea about how prions replicate this process of the two different configurations of the prion, the cellular configuration and the scrapie configuration, and that the misshapen scrapie configuration can somehow change the shape of the cellular protein, making it misshapen, and then having kind of a snowball effect. Okay, that is that for this set of lectures.